Okay, so now we're going to put this back together. It looks like there's some kind of liquid that probably came from the autoclave, so we'll clean that off. I mean, I'm sure it's sterile, you know, because it had to go through the autoclave, but still I don't want any, I don't want stains and surfaces that stuff will grow on. So it's better just to go ahead and clean it up a little bit. Places where, okay, all right. So now we're putting everything back into the incubator. Okay. So you have to remember how this went. These little things face up, so you know that this is bottom and this is top. And of course, once you get it in there, you know which is left and right because it has to face out. So it can be either way, actually, because they're both identical. So I'll put this one in on the left. Have to go in at an angle, and then you can push it up and get it in there like that. Okay, let's go ahead and shut this because it'll alarm if I leave it open. Okay, now the other side. This looks good. And this is bottom and this is top because these openings have to be facing up. And again, go in at an angle. Roll it up like that. Okay. And then the next thing we have to do is insert the shelf holders. Okay, as soon as we get one shelf in there, the sides will stay up. I like to use the bottom one because first it's nice to know how the water bath fits. Because if you use the bottom one, then it's going to be hard to get water in here. On the other hand, it's going to be hard to get the whole water bath out if you need to. Really, there should be no need to take the water bath out unless somebody spills something in there. And of course, you can always pull the shelf out to lift the water pan out of there. But to maximize the space in here, you really want to use all the shelf area. This particular place is in a classroom, so all the shelves are used to their fullest, to their maximum. It's like a New York City skyscraper in here because there's so many cells. Okay, so you take this to where the open part is, so you can slip it in there, and then you push it down, and then you pull it forward. Okay, now this one will fit in. If you push that one down, you're finished. Okay, so you just always do that. You always have one end open, then you slip it to the closed end, and then you put the other one in. Okay, so in, pull it back, and slip the other one in. There we go. Okay. So now both of them are in. We can put one shelf in. So remember I said that the shiny, smooth one is this way, which to me means it ought to go this direction. But then you've got a lip here that you have to lift up to get out. That's why I always have it the other way, which is face down. It's much easier to get cells in and out of here. But now notice that you can't access the water bath. So it's nice to have this water bath pulled out so you can add water and the disinfectant or the sanitizer in there. So having this at the bottom shelf is almost too low. So I might take it out and move it up to the next one. Yes, exactly. Okay, so I'm going to do that. Okay, again, this is very easy to do. And now I've got, this is the open one, so this is the one I can lift up. And then push back and lift that one up. Okay. And then in backwards order. There we go. Now we're on the second shelf.
Yes.
And
No,
that's
it's
much
fine. Yeah.
easier to get in
Yeah.
there.
Yeah.
Okay. So that's how you do it, and then the rest of them, you decide, how much space do you want. I usually, if there's any extra space, I usually leave it in the top shelf because there aren't a lot of tall people.
Mm-hmm.
We don't have a lot of basketball players that take cell biology, so so I usually leave, if there's extra space, I usually leave it on the top shelf.
Okay.
For getting in and out of there. And if you've got metal trays that you can move large numbers of flasks, then I encourage that, because those can also be autoclaved and sterilized and cleaned like the other stuff. So, for example, in older water baths, they have a floor that is a metal tray. And so, if the old water bath goes out and you're throwing it away, keep the tray, use it in the incubator to move cells in and out of the incubator.
Yeah, that's actually a good idea. Yeah.
Okay, that's it.
Okay. Thank you so much.
You're welcome.
Bye, everyone.
Have a good research day.